thanks again for being here.
Thanks so much for having me. It's really a pleasure. And I got to meet some of you yesterday, and I'm blown away by the quality and diversity of people in this group. So hopefully I can continue building bridges between these theoretical and applications aspects. And so the way that this will be structured is that first I'll give a talk that I'll give it probably a bit more slowly than when I give it to a large audience, and there'll be opportunity for discussion. If there are any questions, please stop me immediately. I want to talk through your confusion. And after we're done with that, I actually will pick up where Orion left off yesterday, and we have some new results, in fact, to share. So that hopefully will give some kind of practical aspect to this that you'll be able to take home. Okay, so without further ado, genomes from many frames of reference. I was afraid to put the word pangenomics in the title, so I kept doing that, and I think I'm getting a little exhausted with it because in the end it's biology, and at some point we have to merge our work back with the mainstream of biological research. Just thinking about genomes as a start. This is all probably a little useless in this context, right? So the idea is you've got the tree of life somewhere in an abstract sense, and it's obviously not just a tree because of recombination and so on, but there are changes, mutations occur over time, and a standard practice until a few years ago was you would then be like, hey, I'll take a single reference genome off one of the leaves of this tree and compare everything else to that. And so this is like genome. That's the genome, a genome becomes the center of focus of your analysis. And you extend your analysis. First off, you compare everything to the reference. You extend the analysis by adding a new genome, and you compare it to the reference. And it's by that relationship that you understand the relationship between all the genes in the collection. And so, a very standard approach is that you've got to pass the sequence, your line reads to it, and then you can find the small variants inside of it. So using different modeling techniques. And this already breaks down because you can't easily take this, put it back in that linear string. So you can obviously add more genomes, like every haplotype between the two next sequence. But there's no like, there's just no obvious way to put this back into a string. It doesn't make sense. You can put like, single characters, you can be like, okay, we'll have like an ambiguous base, but an indel that destroys everything. There's no way to represent that. And so this idea to resolve this reference bias problem, as you know, because you've heard like 10 lectures on it or more, 15, I'm not sure how many you have. You build a graph. That's one standard approach. And the graph is nice because the parts that are similar you contrast, and the parts that are different remain. So it's a compressive data structure, an object that represents all these different things. And in principle, if you use that as your reference system, you can resolve reference bias because it's kind of in the middle of all the genomes you're working with. And so this is a pangenomic context. And in that sense, we're looking at the all-to-all relationship of different genomes, which you can think of this being represented this way. So we actually directly understand the relationship between everything. And then when we want to extend the model, we actually either implicitly or explicitly directly compare the new genome with everything we have. And so you can see from this, the number of lines represents, in a way, the amount of work and the amount of modeling you have to do. So it's much harder, but it is unbiased. And that's a very powerful thing in our context, because in principle, we put everything on the same frame of reference and in the same way. This has been introduced ad nauseum. This is kind of fun. It's an early model of the visualization of these of a variation graph. Variation graph is nodes and edges make a sequence graph. That's an old concept that's been floating around since the early 1990s, 1980s. Introduced it. And implicitly in the sequence graph model, there's always this idea that you label the sequences that go through it, but that was never really made explicit as like a formal component in the system until we started working with this variation graph concept. So the genomes you have walk through the nodes of the graph. Here they're described as these labeled walks of node IDs. The colors actually are hashes of some sequence name, and the emoji is also a hash. So the idea here is you could have lots and lots of sequences and keep track of them. It's a hack though, because it doesn't scale quite as well as you can. And there are different ways to look at these. You can visualize them as paths, like kind of ignoring the edges become implicit. Now they're just walks through nodes, which are bits of sequence in a language of genomes that we're considering. And these are representing bowed sequences and any kind of variation between them and such. They're sort of an answer to some fundamental problems in mathematics, because they like let us connect those things in a very rigorous and complete way. Any kind of variation between genomes can be put into this column. And here I'm showing something that's directed and cyclic, but what I mean by any kind of variation is that it's not just SNPs and indels, but also inversions. The graph has two sides. It's actually like DNA. Every node has two sides. Every edge is connecting sides of nodes. And everything implies its own rigorous complement, just as in DNA. And so that lets you represent inversions natively. And when things loop, so if I were to go back through this node multiple times, that represents copy number variation. And of course that's very degenerate, but if I remember where the paths go, then it's okay, because I'm not bounded. It's not like I have a language that can simulate an infinite number of these. I remember all of that. So this idea is so simple and kind of innate. Even as I was saying, when people were talking about sequence graphs, it's kind of implicit in doing this. This is how people think we've been doing it. And if you go to talk to some technical person, perhaps they'll be like, I thought you did that, right? So there's some articles on this concept that came out after the end of the Transitive project. So we said, okay, now we have this map of variation, but we can't really exploit it as a prior and new analysis. So there was work to make genome graphs. This graph theory offered a better way to reboot the human genome. So there's graphs, sequences. This is the model of the MHC, these gamers. This is kind of variation graph type model. And there are several articles on this, including one where these emojis actually came out as like, we got like emojis into popular science literature. And this was discussed on Hacker News, the Big Orange website, which some of you guys might know. If you don't, it's a linked aggregator with comments. And what I was getting at, this is an anecdote, I guess, of what I was trying to explain, which is that here's someone who's probably, if they're technical, but probably not a bioinformatician. And they're a bit angry because it's the tone you get on this kind of site. Sometimes there's kind of like, what is going on? Like, I don't understand how it's possible. Obviously, there's variation. Why did people make it this way? And so on. So, who were the lead developers and what were they thinking? They actually were aware of this, in fact. The Genome Reference Consortium made a graph model because they had the genome that was being built, the assembly, is composite of many individuals, and they had these backs that were sequenced and assembled from many individuals. They made a graph. And in discussion, they said, well, maybe we should release that graph. And some parts and other aspects of the group, other political elements, they said, look, we don't even have the reference, so now you want to jump past that. And so what they did was they did this thing called the golden path, which you might know if you look at these data files. They still refer to this. The golden path is the reference. It's the path through that graph that they chose. And so there's a pangenomic models, right? This is another representation, just for fun. It's a binary matrix. And this is actually very simple because we forget the graph. Now we just have a binary matrix, but I think you can see that it's encoding pretty much all the relevant information about variation, even in this rather complicated SNP indel that opens. So these also can provide a way to simplify analyses, because if you can project into a matrix, you can forget about the graph, then you can apply all kinds of matrix theory and linear algebra methods to it. So it's also potentially a way to simplify it. I won't get into that much. And then because of Italy and visit my adopted country, I can't help but share this. This is not a new idea. And this example, by pure chance, is of an Italian poem. In fact, it's of 9 versions of the poem. And it's showing, for humans, this is actually an archival research study. They will make this thing called a variant graph. It shows us all the variants of a piece of text, so you can understand the many-way relationships, because there's 9 versions, and that's a lot of pairwise relationships to consider. But in this graph, you can immediately see the conserved components of this poem, which in this case so it's about a violin player in a Roman, but it's phrase about a violin player in the Roman countryside. Right? And so it's talking about the bow, this violin, that's key. The author's changing, varying the words that come after that. Another really key word is measure, which has different connotations, but it talks about drawing the bow across the violin, and silence, and of course, the countryside. So, this is something that you would have to do 40 odd comparisons to get a sense of if you didn't make a graph like that. And so this came out before variation graphs. We weren't aware of it until I was putting my thesis into print. Okay, so you've probably heard about PG, a number of cases. Brian mentioned it, and it's kind of a reference point. This figure is actually from a paper led by Tobias Marshall and a consortium of several people in this room as well. That was convened in Leiden at the Lorenz Center, in fact. So the output of the consortium was this description of how we're going to update basic bioinformatic operations to work in the context of many genomes and genome graphs. And so I won't get into the details because they'll be clear just in the presentation, but in effect, anything you can do on a regular sequence, you want to be able to do on a graph. And that includes alignment, variant calling, visualizing annotations, and things like that. And so in my PhD work, I worked with Yoni, and also my advisor Richard Durbin, and a team led by Benedict Patton, who worked at UCSC, to build a basic implementation of read alignment, and actually like the data structures and interface that you need to be able to do applications of these ideas. It's really dependent on Yoni's work on succinct data structure for the generalized GCSA. I'll mess with that, but basically a generalization of FM index to work on graphs. And it's dependent on the integration of these ideas. And we use this, we take this 1,000 genomes cohort reference, which was kind of the inspiration of those journal articles I showed you earlier. We make a graph. The graph is like the VCF, it's acyclic, but it has all the variants in the data set inside of it. And in fact, we don't use all the paths, we're just thinking about the reference as a kind of coordinating system. And we align to this. And so this is a simulation result. We've taken a diploid genome, but we know the haplotypes, and simulated reads for it. And in the simulation, we can track the reference location. In fact, we use the toolkit itself to do that. It's because the reference is inside. So we know the reference location of the reads. Even though we simulate them from a diploid, we can map them back with different methods. Either BWA, which is just against the linear reference, or VG, green against the linear reference, or in red against the parent genome. And the two colors are representing paired-end mapping or single-end mapping. These are the receiver operator characteristics, and they show you want to be as far up to this corner as possible. Right? So that's first off, obvious thing. In general, as you use paired end, you do better. And you also do better in single end when mapping against the parent genome, but not in paired end, because your parent information is rescue the reads. And what's actually going on is that when the reads match the reference, the added information is confusing. And so you do a little worse. There's really no benefit in the case of the parent genome. But for the 10% of reads that have a variant inside of them, that's not the case. You actually dramatically better. You're improving, in both cases, in single end, it's astounding. You're doing like 1.5% better in terms of the F-score. But in paired end, you do the same. And so it just shows that, it shows the kind of trade-off, actually, it's very fundamental, that there's some confusion, you can stop it by making it prior to so it's not all, it's not just improving. And the benefits also, they're marginal. So this is a finding that is very important. And another thing, this is a bit cleaner and clearer. So these are about real reads and genotypes from an actual data sample, not simulated ones. But we know the genotype state in this sample because it's characterized by the NIST Genome in a Bottle consortium. And this is now indel alleles, or actually it's all alleles. SNPs are clear, their length changes 0, and then deletions and insertions ensure the number length. And we have, if we do against the linear reference, we call variance with, so we do alignment with BWA none, we call variance with free base. Then you see an effect where the fraction of observations of the alternate allele, it's about half at the heterozygous SNPs, which makes sense, the alignment's not affected very much. But both for insertions and deletions, you suffer a kind of loss of sensitivity. So reads stop happening because it's not easy for the alignment to see where they should fit if suddenly there's either a piece, of the read that is missing, or there's an additional piece relative to the reference. But for the case of indels variance in the graph that we're aligning against, which is the majority, because most of them are found in this sample, there's no bias for anyone. And here we just have small numbers, so that we see this kind of balancing effect. And then there are some variants in this sample that aren't in the graph. Those are basically rare variants. And for those, the curve, it's even a bit worse than the one for free base and none, but it's similar in the sense that you have a bias as you get into the on the lens. The behavior is very, very similar. And so then we continue to apply this method first on ancient DNA. Or I guess these things kind of happen in parallel. So there's applications to ancient DNA. Ancient DNA is kind of cool because a cool application because you have a really high rate of error intrinsic. There's no way to get around it. The reads are short, and this makes it very hard to align. And additionally having variation on top of that error will make it even harder to see what's going on. And this is as well particularly true in the case of indels. So this is a similar kind of plot. High coverage Yamnaya individual. It's a 4,000 to 5,000-year-old sample from the steppe of Asia. And this shows if you use BWA align, this is about the kind of allele observations of alternate at indels, right? And you see this dramatic fall off. So it's even worse than the 150 base pair rate. It's now short, very error reads. And it's not really the case with the VG graph based on and to dig into this and see what's going on, we went and looked to see cases where there's big differences in the read counts and did some visualizations with this sequence 2 map system. You show the reads mapping across the locus. And so with BWA align against the reference, what happens is there's an indel near the SNP, and they're in phase, in linkage on haplotypes. And so the reads that map those indel, they just don't map at all anymore. As a consequence, we also don't see the SNP. Right? So we submit fewer reads. However, this is a known indel, it's in the population, it's in the graph, we align both haplotypes. Right? This is kind of driving this sort of effect. And this matters for medically relevant loci. For example, this is the CCR5 delta deletion. That this confers resistance to plague and confers resistance to HIV. If we align BWA align against the reference, we cannot see it. Basically, it's invisible because it's close to the length of the reads and so on. And this is just not true with VG. You see the high coverage sample, it's a heterozygote. This is a possible heterozygote as well. And again, this is another one. And we can see this in actual genotyping in these ancient samples. And if you didn't do this with the graph, you could use some specialized method like looking at genomes or something. So you can also use these systems to represent variation just in the population, not necessarily as read mapping, but as a reference system. So here we do Motif scanning. We have a PWM, and so this is the CTCF. This is related to the 3D organization of chromosomes. This motif where it binds pairs of CTCF will produce these looping motifs in the 3D organization that are really important for function. So it has this kind of position weight matrix. If you're not familiar with this, it's basically saying that it's a pattern, and if you saying that in instances where we know the protein is binding, at position 5 we always see a C. At position 6 we usually see an A and sometimes a T and so on. And so this is this kind of matrix model, and we can scan across the sequence and then count or measure like how good a match it is to this particular thing. It's actually graphical. In fact, there's a graphical interpretation. But we're doing it in a different direction. We have the variation graph, and we find all the haplotypes in the locus, and then we just run this across them. We use that to do matching of the motif to the population, which is kind of fun. So then in VG land, the development continued. Benedict taking the lead, and his group did a series of, I should say, his group and me did a series of papers on this topic. So one is about genotyping structural variants. So obviously, if we don't have bias against indels, we can probably do really long indels and still type, and that's effectively what Glenn others showed here. So this is just one example. We have increasingly good data sets of structural variants in the population, and we can build a graph that knows the line reads to them. So again, the kind of read line view. So it reads across a big deletion of an exon. And you can see that you just can't see it if you rely against the reference. It's too big for the microphones. But in this case, we're able to do that. So we can do genotyping of SVs, of known SVs, quite easily. And so then this idea got taken even further in work led by Yoni, actually a lot of folks who invested a huge amount of effort on this. John, Shen, Adam, Jordan, and in effect, they write a new alignment, which I guess you described already.
Yeah.
Maybe you presented these already. Yeah. Okay, good. So you know about this, and you can see where it's going. And yeah, we'll also describe this probably. So this is just highlighting the way the sequence of the graph alignment occurs. So in generalized sequence alignment of graphs. And you show the results as well. Okay. Okay, I won't belabor it. Basically, they make VG kind of mapping practical. They're making it fast and more than good enough to do really cool applications. This is just about the runtime. Show this. You can do variant calling. You can reproduce the result we had from before, but it's faster than BWMM, and it also lets you do this cool population genetics application of looking at the PCAs and different structural variants, and they look like the ones you get from the steps, basically, which is a really nice confirmation of something we expected to see, but wasn't so easy to look at. Okay, so anyway, we use these things, right? And you guys have seen that. It's shown you, and others have shown that it's possible to use them. But it really remained hard to build them until the past few years. Because one problem is just the data availability. We had short reads and a reference to find the variants, and make a graph on it. And that wasn't sufficient to really build a complete kind of graphical model. The idea is that I was describing, right? We just have this directly cyclic graph with bubbles. Okay, so please see some insights. All right, so there are many ways to do this. And you might have seen there's another common technique, which is a progressive alignment using a tool called Minigraph. I don't think in these slides I really get into that much at all, but I do want to describe the method that I've been working on called the Pangenome Graph Builder. It's not just a specific method, it's not just one executable, it's actually a workflow. And I think that workflow, it suggests some things about how we can do this in general. It provides a kind of standard for future graph building methodologies that, in effect, you have steps that build a lot like this. You build an alignment between sequences. This alignment implies a variation graph. So you can reduce the graph out of the alignment and the set of sequences you have. But this is a disordered object in this case, it's deterministically ordered by order, or the latent structures in it are not obvious, right? So the topology is unbiased with respect to the input, but we can't really tell what's going on inside of it without learning things about its topology and sorting and organizing it. So we do this organization, and this then allows us to do refinement and normalization processes on the graph. So every step is actually reading standard data types, like this is taking a FASTA file, and then this is taking a FASTA file and an alignment file in path format. And then these steps, they work on top of the graph file in it's called GFA. So the point being that the graph then becomes like just a primary object of analysis, and it's modular. So if you have a better way of doing the sorting, you can plug that in and improve this kind of process. Just as for short read mapping, if you had a better read mapper, you plug it into your pipeline and you do better. Right? Or if you have a better way to sort your reads, you just change it, and because the input and output is the same, everything just gets better. So that's the basic model. And this is it's built on a few concepts that were necessary to scale the approaches to whole genomes. So the first problem was, and this is in some sense what minigraph, has minigraph been presented? But maybe I should do that later. I have some slides on if you guys want to talk about. Okay, anyway, a progressive alignment approach, where you align a sequence to another, you make a graph, and you align sequences to that graph. It kind of deals with this issue that the scale of alignment between many whole genomes is really, really big. If I take like just a few copies of one chromosome and align them to each other, I have an exascale, matrix that I want to consider. And so that's a big issue to try to deal with. So to deal with it, we built a new alignment algorithm. It's called WF-MASH. It stands for Wavefront Mash Mapper. There's a mash map is a technique where you have segments. You're not mapping k-mers, as you would with mini map 2, when you find a bunch of k-mer matches that chain together. Instead, you go to a much larger scale. So you work at the scale of 10,000 to 100,000 base pair sequences, and ask how they relate to each other in a k-mer metric space. So you have k-mers for both. You have a subset of them. They're called minimizers. There are a predetermined subset of those. And you can use them to make a small representation of the larger sequences. And then those get compared to actually do the mapping. And then those mappings, they get chained together into longer sequences. So I'm not presenting that part here. It's actually prior work building on top of it. But the problem with this is that you get an approximate mapping using two sequences that says, from here to here at A and here to here at B, they should align. But you don't have any information about the exact k-mer matches to guide the base level alignment. And so we had to develop a new base level aligner that was capable of working at the scalable chromosome. And so to do that, you don't want to do Wunsch, for example, or Sniff-Waterman, where you kind of blindly fill the matrix and then trace back through the high scoring or best scoring trajectory through this matrix to find alignment, because this is quadratic. And just like now we have an exascale compute, it's just not conceivable. Instead, several things have to happen. So one, I mentioned, is heuristic. We find the region we know by homology. And then we apply a new formulation, an integration of a lot of ideas that have been floating around in sequence alignment for the history of sequence alignment. It's called the Wavefront algorithm. And it reformulates Needle and Wunsch as a progressive exploration of cells the matrix can reach at a given score. And you do this progressive exploration until you get to the end, at which point you don't need to do any other computation. So this and this actually they provide exactly the same result, but with a dramatically reduced amount of computation. This is in order of the score. And the score is related to divergence. So it's something like divergence squared, rather than length of sequences squared. And that's great, but when the sequences are really big, or the divergence is really high, it's still really high power. And so to actually make this work on whole chromosomes, we do another layer. We actually make a high-level version of this. Yeah.
So what are the theoretical guarantees about this to work?
Of this one?
Yeah.
This is the same guarantees as in Wunsch. It's an exact algorithm. Yeah, 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 it's exactly. So it's giving you exactly the same alignment. In this formulation, no. So you can do something where you reduce, you remove some of the parts that are very unlikely to succeed. Then you lose the guarantees. But if you don't do this, it's called reduction. So if you were to say you're here, and you have a rule that says, if I have some wavefront that's expanding, but it's more than 50 base pairs in your line, that stops expanding. Right? It's kind of an adaptive banding.
Yes, it's a banding.
It's a banding technique, yes. If you do that, it doesn't have the theoretical guarantee that you get down more, because you could go outside the band. An optimal line might fall outside the band. Yeah, it's not a fixed band, actually, it's an adaptive band. But yeah, the adaptive band can fail. So it provides the same theoretical guarantees as a full evaluation of matrix. Yeah, and then this idea has been floating around in different formulations for 20 years, 25 years, even more, 30 years. G. Myers and stuff, it's very like this. Dijkstra.
There are older works on this wavefront.
Yeah, they're all hinting at it, but somehow they didn't get integrated into a gap affine module in like the library. All the synthesis of these things. And also just calling it wavefront, I think, is helpful because it clarifies like it's this expanding exploration that is in fact really key to the process. And actually, that was inspiring. So as I was saying, the score gets really big. It's still quadratic, so it takes too long. We can't just apply this whole chromosome. And instead, we apply it to not base pairs, but cells of the matrix that are like 256 base pairs. And so this drives down the order of cost by a kind of quadratic factor. It's like 256 squared, right? Less compute in the big O sense. And really, it's difficult. So you guys are used to this now, right? Okay. So this is showing an alignment of 2 E. coli genomes, or a few megabases of them. And what's being shown is basically the way that this works. Rather than looking at single base pairs, there's a callback function that says, look at these two regions of these two sequences. And if the Jacquard of the Kamer space is above some threshold, then we attempt an alignment like this one. And if this one succeeds at some very loose threshold, we say it's a match. So it binarizes the comparison of these two sequences. And then it behaves the same way. And so here we're actually showing gray cells are where it doesn't match, green ones are where it does. And you can basically see the optimal alignment on this thing. Okay, so that's a way, shown visually to align the chromosomes to each other. So given that, I said a set of alignments implies a graph, and this figure attempts to describe that in detail. It's pretty straightforward, and this is not something like another one of these obvious things that just needs to be synthesized, and it's existing in a lot of other toolkits like the Cactus Genome Aligner, for example, other alignment systems, even multiple sequence aligners. They're doing something very similar. So the idea is up here we've got a text for genomes. They have characters, right? They're DNA bases, and they say Gattaca, because why not? And here, these represent alignments between them. So in the alignments saying that's the same thing in some evolutionary sense. They have a common ancestor, right? And they're the same characters, so they exactly match. So we've got these alignments and between the sequences in the dots. So the algorithm, the sequential algorithm, progresses by starting at the first character of the text and following all the alignments to transitively relate characters in the text to each other. This transitive closure, or a union find output, implies a single character output graph. So in the simple case, the graph, one character, one node, is what I'm showing here. These would make the first node of the graph, right? And then continue. Next one, only these two map together, so we go there. Continue. All three T's map together, and so on. You can see where this goes. Now, we remember the mapping from these into the graph. And also we remember the mapping, remember which characters have been used as we're progressing. So if we get here, we're like, okay, we already close that into the graph. But C is new, and so we would add it as a new node, right? And so this process builds the node space of the graph, the sequence space of the graph. And we can reconstruct the paths using the mapping I was referring to. So we remember where we've projected this into the graph, and we can walk through to make the appropriate path level. And then the edges are just telling you what the paths are. And then there's a final step, which is that you can compress a run of nodes like this. If I have the same input and output paths, then I can just compress them into one of them. That's the method, and it's implemented using what we call match compression, but it's just using ranges of matches rather than single character matches. So here, this encoding is kind of implying that I've got an integer here saying it matches to another integer. It's pointing into my set of sequences, but really, we work with like a longer matches. Say there's an exact match of 1,000 base pairs, right? So I compress the representation with respect to the divergence between the sequences, and then that gets encoded in implicit interval trees that are just sorted arrays of these intervals. We can ask questions like, given this, I'm in this position on the sequence, what is mapping to me? And then that actually evokes this process. So this back and can scale to like thousands of bacterial genomes or hundreds of human ones, and it lets us, and it's also really dumb. So it just takes the input alignments you have and makes the graph completely structured to fall to the graph based on your alignments, which is kind of a nice thing for users, application developers, because you don't need to think about the graph construction algorithm. You just need to think about the relationship between the sequences you have. Okay, so then, on zoom in's a little slow, but in effect, given that graph, it's disordered because it comes out in this kind of order that we see the novel bases in the sequences. And we need to sort it. So we learn using stochastic gradient descent type model, an optimal layout in one dimension for the graph. And then this optimal layout, it's guided by the path distances. So it's kind of representing mutual information in all the paths. We can cut this up and locally run a partial layer alignment, a multiple sequence alignment on each part to compress them more tightly. So if there are problems with the initial alignments, we can resolve this in this normalization process. And then finally, this is fun. The layout also works in 2 dimensions, which is helpful because we can do things like do a 2-dimensional layout of a graph of MHC, humans. So this is an important locus that relates to immune response and self-recognition. And then this toolkit, OGG, is, it's funny, it's published now in bioinformatics, but I don't have the link to the bioarchive still. It lets us do things like visualize, but also cut out parts of the graph. So it does operations you might expect out of something like embed tools, but to generalize the work on the graph. And it uses the paths in the graph as a coordinate system. So you can have descriptions of ranges over the paths. You can use that to cut the graph. You can use it to ask, like, what's the path depth in these ranges. You can use that to get a visualization of parts of the graph. And you can project out pairwise alignments in the graph that show you things like what's happening at this locus. This is a gene called C4, and it exists in different copies in different human haplotypes. It's interesting because it's related to schizophrenia risk and other things in development. And so if we were to cut that out of, it's in NHC, so we cut it out of here, we can get a graph out. We look at that in another tool called Vantage. Right? And OGG can give us labels to color the nodes based on this is where the C4 gene is, in green. And we can zoom in and actually see the difference between different haplotypes. These are the SNPs that distinguish the 2 versions in human reference, C4A and C4B. The visualization also gives us this kind of matrix. There is a thing called OGG Viz. It gives us a matrix view. This one is just showing you where the paths sit. This shows you the progression along each one. It's kind of implicitly showing inversions. It's a bit messed up here because there's a loop. That's the key thing. So genomes are going around this multiple times. There's one copy of C4 in the graph, but you could have multiple different genomes. And this is showing inversion. This is showing the depth. So here, gray is one fold, and then there's a rainbow going up to very high coverage. So there's 1, 2, 3, and so on. And You can pull the alignments and plot the graph out with an algorithm called Gaussian tangle. These will show you things like relative to CHM13, the T to T assembly. You can see which gene is deleted or duplicated in parallel extension. Actually, I have other cool stuff on that, but I forgot to include this slide. So anyway, this last part is applications of these techniques to the human genome, which is a really, really great data resource that's being built of eventually hundreds of diploid assemblies of human genomes that are effective in whole chromosome scale. So we have a first release of this called the year 1 freeze. And in the year 1 freeze, there are 44 individuals. Some are held out, I'll just say there are 44, plus 2 references. We have CHM13, which is this new assembly, complete assembly of the human genome, and GRCH58. And for our sanity, they're partitioned by chromosome. We have a mechanism to pull these into different chromosome components and work with them individually. So you could put them all into the algorithm at once, but you might need a larger computer or more time than one weight, harder to debug, and so on. So we broke it into pieces. And actually, in the consortium, there are actually 3 methods being applied. There's the mini-graph method, which is a progressive construction of a graph based on a reference that is designed to capture structural variants, like novel large sequences. Then you can add base level variation to that with the local multiple sequence alignment. And then the alternative is what I presented, what I've been developing, the Pangenome graph builder. So we make all 3 and actually use them to do cross-validation. And so this is showing kind of cross-validation with both the mini-graph cactus and PGDP graphs that are base level to look at SNPs. This looks at small variants, things less than 50 base pairs long. And they've been detected, to make a truth set, they've been detected in alignment to the reference with the input hi-fi sequences, the hi-fi sequencing data was actually used to make the assemblies. And then that gives us the kind of truth set to compare what we get out of the graph. So we can make that SNP calls out of the graph, compare them back. And I just want to point out that most of the data is in what we would say is easy regions. What's that called? Most of the data is in easy regions. And in those regions, we're getting extremely high accuracy with this cross-validation method, above 99%. And that's consistent in both of the methods for graph building, actually. And this is basically true in things where we think it's doing all right, but even works okay in more difficult regions of the genome, like the C4 locus. It's duplicated. And actually, yeah, I'm going to go a few more minutes and pause for 5 minutes. Right? I see folks are drifting. So, is that okay? Or maybe I can, there's about 10 more minutes of this. If I do that, we break, and then we can do practical after. Is that okay? Okay, so hold on tight. Anyway, this is just showing the same for structural variants, that basically we're able to bind structural variants in the graph, and it behaves quite similarly. When things are easy, we get 99% precision, and this is pretty consistent across the length distribution. It's showing the length, insertions, deletions, and this is the precision recall. It's consistent. It's good. And yeah, again, these graphs, they let us gain insight visually, almost like a microscope in the pangenome when we look at variation in complex loci. This is the RH group determination locus. In the mini-graph cactus graph, it shows basically there's an additional copy of RHD in some samples. You can pull the haplotypes out and draw them into these gene arrow plots here. These are different haplotypes as they walk through the graph. There's an inversion, it's kind of neat. This is the HLA-A region. There's HLA and a bunch of pseudogenes of HLA, that some of which aren't functional. And you can have insertions and deletions of different of those. This is in the PGB graph. So, yeah. It lets us look at these parts that are hard to see. And we can also look at whole chromosomes and think about the chromosome scale patterns of variation. So although The alignments in the centromeres, there's a lot of controversy about whether or not they're working. We do see a kind of VNTR, variable number of tandem repeats at the centromere, which matches our expectation. There's high order repeats that are very similar. So each genome is looping through this some number of times, depending on the length of the centromere. And that's this is chromosome 1, and these are different genes, so there's 20. Yeah, but most of the euchromatic regions of the genome are very simple. They just look like a Y. In fact, they are very basic graph in most cases, except for these segmented regions. So this is a kind of cool spot. It's a locus on chromosome 8 where there's an inversion. It toggles back and forth. There's a paper that's recently shown it's probably very frequent, even at a rate of like 1 in every 1,000 or 10,000 births. And the cause of that toggling is actually these repeated genes, repeated cassettes, in effect, around the base, or the flanking regions of the inversion. They can recombine using non-allelic molecular recombination in such a way that the sequence in the middle flips around. And so this shows the two references relative to each other. It shows this kind of untangling of the graph that lets us look inside of it. And so this inverted sequence, it corresponds to this region here. These are the SNP density across chromosome 8 in the two references. So we can just look at the top one. The color shows if you're nested inside another variation. So as the color becomes more spectral, you're more nested. And this is the inversion. The thing that's neat about this place is that this whole region, not just inside the inversion, but around it, actually has higher rates of variation than the background of the entire genome. It's one of the highest regional rates of variation. And this probably would be consistent in other species that have these kinds of things happening. And then, there is apparently some kind of functional advantage to this in the sense that the genes that are in these polymorphic regions near the boundaries of that inversion, they're defensin proteins there are many defensin protein genes. These are effectively antibiotic type genes that provide antibiotic resistance. And not resistance, sorry, they're weak. They're antibacterial, and they're secreted in the human epithelium. And then, yeah, it's slow. Okay. Zooming in on this knot at the base of the inversion, there's something kind of cool. This is repeated a huge number of times in many different genomes, huge number, like very, very, very high rate of variation in length. And then the copies, it's called FAM90A, and we don't know what it does. So there's like, this is highlighting things that could be interesting to look at that are poorly understood. Okay, so then it's getting a bit long. Yeah, I guess this is kind of the stopping point. This is the MHC. Again, I talked about the same locus before in detail. This is showing annotations on top of the graph. And again, showing the same locus that I showed with the Augie bits, for example. And I think even the computer's getting tired. So this is just as a point, because as a question often comes up, this is different species in the graph at the same locus that I was showing. These are primates and human genomes, and this shows the same basic structure that you see. One really funny thing is that the humans here, they appear to be closer to orangutan than to chimpanzees. So in the MHC, it's actually rather common that what's called incomplete lineage sorting. The divergence goes back 40 million years. Okay, and that's a good stopping point. I have about 10 more minutes of slides, and then about 40 minutes of interactive stuff. We can do what we talk. But everyone looks a bit overwhelmed, and hot. So maybe we stop for 5 minutes and reconvene. Is that okay? Super short break. Okay, so at 12:05, we'll start again. Okay? Oh, I just told you.
Maybe that's.
Okay, the last part of the lecture, that's I know banging on your brains, is to give an example of how these kinds of models will let us understand things that weren't visible before. And so I'll get into that. It should easily fit. And there might even be a chance to do a response to Ryan's example from yesterday. It's fine. No, it's fine. Let's just go. Okay, so we finished the human genome, right? I mentioned this before. There's a CHM13 telomere to telomere assembly. It's super cool. And yeah, I messed up these arrows. Actually, they should be like a little bit further up. Basically, we're filling all these black parts of the human reference, or incomplete. And this telomere to telomere assembly uses new sequencing technology and random assembly methods to fill through them. And so we close the centromeres. This one, chromosome 9, for example, is really big, and these arms of the acrocentrics. That if you play in human genomics, you know they're like ends, just a bunch of ends. I always wondered what's going on there. And in fact, we begin to see what's happening in these places. So these short arms of the acrocentrics, that's where the ribosomal DNA is, and that those ribosomal DNAs in the assembly graph that led to this. See them as these tangles here. And the thing that's interesting is that all the chromosomes in this assembly, except the acrocentrics, are isolated components in the assembly. And that's very interesting. So there's some discussion about what's going on in this paper, but I'll take a look at it through the lens of the Human Pangenome Project. But we have a lot of assemblies on these acrocentrics, and we can look at them. So first thing that kind of came up in the Human Pangenome Project was that we did an assembly evaluation, and one thing you do is you take the contigs from the assemblies and you map them against the reference, against, in this case, the telomere to telomere assembly. And what showed up was that pretty much all samples had what were called misjoins between different acrocentric chromosomes with respect to the telomere to telomere assembly. So this is basically like, a contig here is mapping a bit to both chromosome 15 and a bit to 22, maybe, or a bit to 20, 21, right? And this is a mapping graph. It's showing us how the alignments of contigs are to the other contigs, which are, in this case, the chromosomes of the T. And this is really interesting. So, yeah, it's just lagging. Okay, it's fine. So then we did this with all the contigs to all the other contigs directly, and so it's not reference added, now it's reference free, and the reference ends up just colored in this, and it shows basically the same kind of pattern. You have these tight associations between 22, 14, 21, 13, and 15 is similar but a bit separated. You can apply a community detection algorithm to this mapping graph, and the communities you get out, in most cases they just have two reference homologous chromosomes in them. But in the case of these two clusters and the sex chromosomes, which are linked together by a homologous region at their ends, you get a community that binds them. And so the community is basically saying that it's the connectivity of the graph implies that these are it's better that they're considered as a unit in a way. So then, sorry, there's like 5 seconds of latency now. So okay, I'll just have to go ahead. If we take the context mapping to the T chromosomes and build graphs out of them individually, you get this kind of pattern. The Q arm, the long arm, is, as we've seen, pretty simple. These are the centromeres. In the short arm, and leading up to where the RDNA is, things get really crazy. And they look a bit like puzzle pieces that should fit together. And so we did actually put them together in a single graph. And this shows that intuition is correct. Actually, there's really strong homology between parts of that short arm on the different chromosomes. So you get this kind of dancer, I guess, up there in its body is it's this sort of one single representation of the short arm of an acrocentric. And there are these different permutations between the various chromosomes. The pattern is that 14 and 22 are closer together, 13 and 21 they come together actually before the centromere, and 15 starts to link into the system further toward the rDNA, the ribosomal DNA on the short arm. And this, of course, matches the assembly, the result we have in the assembly. So, yeah, so then we've redone this. And this just describes the process again. So we take long contigs, in this case, de novo mapping. In this case, we're going to label the contigs by which chromosome they come from. It should be a little bit easier to see what's going on. And we'll do this kind of community structure analysis again. And this is just a bit more detailed. It's actually showing where the sex chromosomes are as well. And also, it's showing the active chromosomes. If we were to zoom in on this, then we get this kind of, it's fast again. We get this kind of pattern. It's quite similar to what we're seeing in different graph models. But here again, the nodes of the context, the coloring, the part where we think we should go. What you can see is that in this middle part, it's just a big mess because we don't, these chromosomes, maybe the assignment to chromosome is not even possible. Right? And so we basically look at just the best assemblies in these regions. And we do that by, we wanted to have context where we were pretty sure which Q arm they correspond to. Like that's the way we can make an assignment. And so we take all the context from HPLC, these nice haplotype resolved assemblies, and we map them, and we find the ones that cross the centromere and are deep into both the Q and the P arms of the T chromosomes. And then we build a graph from them, because that's our technique. Okay, this is old news. And the graph of these particular ones that are very long, and so in effect kind of higher quality, it looks exactly the same as what we've seen before. This is a visualization of layout algorithms in Logi and a tool called GFaces, which allows us to label the arms with these colors. So we have actually a bed file that says like what color that ring should be, and so we label the different chromosomes. The lengths are sort of approximately correct. So for example, 13 is much bigger than 22. You can see that. Then zooming in up here, in this region where the tangle happens, we see the same pattern, right? So 21 and 13 come together, 14 and 22 come together, and then 15 hooks into this sort of satellite tangle deeper. And then finally, it's interesting to see where like the RDNA array is. We actually see a little bit of the edge of the RDNA array. We can't assemble through it, but we can look into it. Another funny thing is that these are the ends in GOCH38. They don't map anywhere, and they're just tips coming off the ground. They're still in there for fun, basically. And okay, so then what's going on in this mess? Right? The graph gives us a hint, but we want to know what is there a process that's actually driving this, or is it just some kind of ancestral homology? And we can do that by looking at patterns of recombination. The way we do that is a kind of chromosome painting. So every contig is going to be painted in effect by which TT chromosome it maps best to at every given location. And we do this with something we call untangling. Untangling, I mentioned before, it's taking the graph and it's converting it into pairwise alignments. So in this case, we have the query, only these target reference chromosomes. We segment, an algorithm will segment the graph. And then in every segment, we could use a Jacquard metric over the allele space, the node space of the graph, to make a mapping, in effect. And so this will say, this is like a multi-mapping, it's saying query from A to B matches to target 1, from here to there, with this Jacquard. So it's exactly the same as the red, right? And it's a little bit different than the yellow one, and it's very different than the blue one. Right? And then to make this make sense in the space of a reference, you have to do the second step, which we call rounding. And so this will mix it with something where we're forcing an alignment to one of them. So now we want to think about this system relative to chromosome 14. We could do it against any chromosome, but in this case we'll focus the analysis so we understand where it fits in the space of chromosome 14. And we do that by just doing the entanglement, forcing it to map to this particular one. And then finally, we're going to plot. So we're going to use this information to make a plot that shows what is the entangled identity, like what chromosome are you painting at every position relative to reference chromosomes that are inside. So this is like head exploding kind of stuff, but basically here it's saying like this contig, it looks like chromosome 14 for a while, and then in this region, that relative to chromosome 14 is here, it looks like 13. And this gives us access to understanding what's going on inside of that tangled region. This is the PR, so we're going out to 25 megabase graph. This is a genome browser visualization against T to T. The rDNA is here, the centromere is here. This is the key one. So in the centromere, the entangling doesn't work very well, the alignment doesn't work very well, and part of the reason is probably the assembly quality. The Pi assemblies are not super good across the centromere. They do okay, but they're not perfect. But this region here is also suddenly a repetitive sequence, but it's in a kind of one-to-one copy state between these different chromosomes. And the entangling shows us something quite interesting. That basically, so first up, these are really nice assemblies that are based on also nanopore data in addition, so we have high confidence in them. And they basically show a pattern of matching between 13 and 21. Right? And we also see that in other contexts, and we also see stronger matching to 14 and some. Right? So that these patterns of recombination in effect between the rDNA and the satellite sequence. And the centromere sequence. And we can aggregate these, right? So I just showed a few contexts, but if I go across all the contexts and I count how many in each region are mapping different chromosomes, I take these curves, one for each chromosome. And so on 13, we're looking at the space of 13, you have almost everything matches there in the Q arm. And as you go to the centromere, the satellite similarity between 13 and 21 is shown here. It matches to that. And then in this particular region, you get a mix. And that mix, it actually corresponds to things seen in the T to T paper. That you can look just at the T to T chromosomes and you see the same kind of technology. This mix corresponds to these red bars that show the relationship of 13 to the other aprocentrics. So we're seeing that they're very similar to 14 and the 21. And that's found in the population as well. It's not just a feature of the T to T. Yeah. And then the final thing, this is kind of a way to get a quantitative metric of the rate of recombination. We can measure the entropy of this untangling in Windows. And you see, in effect, that it's increasing as you get toward the RDNA. And so this suggests that the RDNA, homology between these repeated 40 kilobase copies of genes between the different chromosomes is allowing for recombination. Driving process of recombination also affects the surrounding regions. It's not just isolated to the RDNA itself. Yes, okay. And then just I guess this is to drive the point a bit further. So those previous plots were the first best mapping, but now just to think about the multi-mappings as well, to show you that it's not just, there's similarity between them. And so here we're showing the 5 best mappings for every config in a set of like 5 or 6 configs. And what I want to draw your eye to first is this region here. Here, there's homology between 21 and 13, but it would appear to be ancient because the pattern is always that the Q arm rounded matching configs. They just have homology. They don't match better to 21. So it's like implying it's not an ongoing process of recombination. It's some ancestral thing. But that's not the case here. Here, you see this flipping. This pattern is what you see in the pseudo-autosomal region on the sex chromosomes. And it would match basically our intuition that there's recombination. So they're very similar matches, and the order in which this painting is happening relative to the config is different. And it's not just a random mess, although in some there's quite noisy assembly. In particular, the high quality assemblies based on nanopore and high you see plots that are of the scale that would be consistent with combination. And yeah, you can look at this from other frames of reference as well. You can look at it from the frame of reference of 14. Here you see this relationship with 22, it's purple. The same pattern, that there's this increasing diversity entropy metric as you get close to the RDNA. And it's also true to some extent of 15, but as we saw before, there's 15 is a bit out of this kind of complex relationship. And yes, this shows that on 15, most of the action is actually very close to the ribosomal DNA. In fact, there's some kind of recombination happening there. And then this is 21. So like looking back, remember we were looking at 13, so now we're kind of seeing that same effect from the other side. And again, it shows the same kind of pattern. Again, the pattern of increasing diversity entropy approaching the RDNA. And then finally, the same is true on exactly the same kind of pattern. So, and then this is just a technical note. So to have some confidence in what we're doing, we have assemblies of the same sample, HG002, both just from HiFi and from an assembler called Virgo, actually by Miko, who visited us yesterday. So this combines the HiFi data and it makes a graph of the HiFi data and effectively untangles that, like our pops bubbles and complex regions using nanopore data. And so it's described here, and we can compare it to the HiFi assembly, which is broken in context. In most cases, you see some concordance. In a few small cases, there's confusion. Although, I mean, I think given what we're seeing, it's understandable that some of them might make a mistake in such a region because there's such strong similarity between different chromosomes. But in effect, patterns like this recombination of 15 to relative to 15 are maintained. So this gives us some confidence, and of course we do this for all the different chromosomes. It's mostly concordance. And so in effect, this shows us that with these really amazing assembly research and these new analytical techniques, we're able to dig into something that a question that's basically been open since 1988 when the satellite sequences of these were first understood. So it was obvious that there was very similar satellite sequences in the centromeres, and the hierarchies were similar, and so the researchers here suggested an ongoing process of recombination exchange. But to verify that, you had to have assemblies of these regions of many individuals. And so we hope that we're basically closing this question, at least clarifying what's going on as part of the genome that until a few years ago we all saw just the string of nodes. So, cool. Yeah. All right, that's it. Should take a ton of people. I'll do this for a second. I guess we have about 5 minutes, and in fear that perhaps there won't be an opportunity tomorrow, I wanted to share something that Brian and I were looking at. So yesterday, you guys see the screen enough? Okay, so yeah,
Right.
yeah.
Right.
Can I go 5 minutes?
No,
Okay.
no problem. Go ahead.
Yeah, we could have a longer practical.
Yeah,
but-
question.
Oh, excuse me, John.
Yeah, sorry. Maybe like from what a lot of Tango does, a lot of point was like to break cycles or to move erroneous source.
Yeah, but
from what you described, it's like it's doing something like some local alignment, like blasting it, and also the other thing is that if you're only checking the card, would it you get tripped up by any random computation of the bases?
the last question, I don't know. I think we can sit down on some paper. The first one, so the early presentations of this, it was like a way to understand repetitive sequences. So in that sense, the segmentation is what you're thinking about. It's segmented so that the segments that are being compared are linear. The segments don't themselves loop. And so that we actually have an algorithm in the segmentation that looks for loops and then cuts them to make the segments be like linear in sequences. And what it's doing is it's taking the graph and making an alignment, in effect. Yeah.
So a tunnel is not like mapping, so some kind of...
It's like querying the graph and saying what's the alignment or really mapping between two sequences, or rather one sequence and many other target ones, which could be one, it could be the whole set. So it's like gonna let me express like a genome as like a bunch of segments in other targets that I specify. Good.
There was Tosh?
Yes.
So thank you. Finally, a presentation for biologists. I would like to ask, how about private mutation in case of Pangenome graph built on 1,000 genome projects using short tree?
Private mutation.
Yeah.
Like if it's not in the graph, it doesn't get any easier. That's the real problem.
Can you let's say like flag areas where it's a twilight zone or
I guess you get a benefit from the fact that you can know if it's been seen before, but that's the real benefit. You just know what the background is. Your prior is more complete.
yeah, when you are doing like resequencing,
Yeah.
on Illuminus, you're looking for the private mutations.
Yes, yeah, but you can be sure that if it's in the graph, you'll match it easily. So at least that will help you distinguish between private because it really is private or private because you couldn't find it. You know, that's before, right? So, and I guess that's as best as good as you can do, though. Because the graph is like a private. If you haven't seen it before, you don't get any benefit from
But
it.
is there a way to, let's say, flag at least that there is a twilight zone?
I guess. I mean, if you aggregate it across a lot of samples and you see noise somewhere, you see like in effect these receptor PRs in the twilight zone. We don't need to do a good job of doing variant calling on them in the graph. You know, yeah, you could imagine these kinds of things coming out of the graph model. Any other quick questions? Or long ones?
Okay, since there are PhD students here that are interested in open problems, a question is about the current VG suite. I mean, what are the main limitations in your opinion? What are the main open
VG? Yeah, okay, so.
VG?
Okay, VG, the main limitation of VG is it's a monolithic software package like
Yes,
in C++.
I know.
So that's a big limitation. We should probably make independent tools that work on graph data models like the GFA model or other ones we might come up with, but on the data model itself, you know, not inside of the massive umbrella project. And documentation of it. That's suffered again because of monolithification. Small things are easier to clarify. So that's a big problem. If you're thinking about making algorithms, try to make them in an independent way. Another big open problem, this is kind of an issue, it's that many projects, they tend to solve a subset of the model space. So they say, well, this will work on a DAG. Okay. So then you develop something that works on a DAG. Nothing I've shown you is a DAG. No biological graph that's large enough made in this kind of way would be a DAG. You know, so I think people need to be, folks who are working on these kind of methods and just learning about them, need to be sensitive to the fact that those limitations may seem practical because they might make an algorithm practical, but they become impractical because then you can't actually apply them out to real world data. Or you're distorting, you have to distort the real world data in some very strange ways.
In your experience with all the works you have done related to human pangenomics, what are the main issues said about the alignment of reads the pangenome?
The reads the pangenome. Well, short reads are short. That's an issue.
No, in general, alignment of the pangenome.
I mean, I guess the cutting edge is more like we're able to assemble really repetitive sequences, like the ones on these genomes. We can't assemble RDNA really easily because it's very big, very long, repeats, but now we can assemble centrifuge. And so it's a little unclear if the methods we have are doing the right thing, aligning through those. And some folks actually say it's not possible. And there's current methods are just completely broken. So that's kind of an open problem. And in issues of aligning to the pangenome, I think the issue is that there's an entangling of the graph complexity and the alignment operation. That's problematic because the graph complexity just relates to evolution. And that, again, I was thinking of that in the last comment. It'd be nice to be able to make a graph however you want and not have it affect your alignment performance. Like the alignment can still be practical even if the graph represents complex patterns of variation, loops and tangles and big bubbles and these kinds of things. And that's something we don't have, actually. So to align reads against these graphs, I haven't even tried to align against the PGB graphs. And to align against any of the graphs that we're actually allowed to work with, even the mini graph practice model has to be pruned and modified, even though it was explicitly designed to make it easier for you to align. So we need methods that are comfortable with the complex mess that's out there in the world, or achieve some kind of balance that makes it so that it becomes invisible to the user. Because it's just not. I don't think it's acceptable if you say like this should be a model of comparative, genomics and evolution. But then, okay, now to use it, I have to like make a bunch of crazy assumptions about like what really happened in the past that aren't true. Right? So I think that's a really big open problem. If you're giving me 2 minutes, I'll show you a response to Brian. We had a lot of fun during the break, and I want to share this. So yesterday, Ryan showed us examples of two equalized genomes. And I think the first problem was, well, it's not a problem, but Ryan is building an understanding of this process by working with individual tools. And so the parameter space of them is very confusing, and they don't have good defaults and so on. So the WF MASH aligner is kind of an independent thing, and pretty works well on its own. Sequish, let's say, as well, basically the case. But the SmoothXG, the graph normalization steps, visualization, all that is very hard to orchestrate. And so it's in PGDB, which is a shell script that organizes it according to a few key parameters. The key parameters are the number of genomes. It's probably the only really important one. And then there's two other ones that really affect the graph structure. One is called the segment length. It's like the size of that sequence you do the match mapping with. And the other is a percent identity. It's the average nucleotide identity limit at which you make a map. So I have PGDB. We have a new release. Of course, that wasn't the one that I was working with, so it's on me. But it has, I guess, decent default settings to work with this case that we have. And so it'll take a few minutes, so I won't rerun it, but it takes about 2 minutes. We took the genomes that Brian was working with, and this is actually, I'll just show you this is the command line. So it just takes the fast as input. It says this is an output directory, I just called it X. So it's lazy. There are 2 genomes, and I only use 16 threads. Unfortunately, I have 7 threads. Probably should automatically detect it. But it ran, I can tell you how long it took to run. So something, no, I can't tell you because there isn't a total thing. But it took less time than it did to go downstairs and get a coffee. And it makes these visualizations that are kind of explanatory. So you can't see this. It's completely linear. I'll show you that in a different way. This is this AlGViz. It's like a binary matrix. These are the 2 genomes of iron. And they're actually completely collinear, in fact. There's no inversion, which is what we were expecting to see here. And so we saw this, so these are these kind of visualizations. I can also show you another one. It's called so this is this interactive visualization tool called Genomes. And it looks really jerky from Zoom, but it's actually completely smooth. And you can, so you can zoom in and like select nodes, and then you look at the nodes, and you can say, okay, there's 2 paths there, and it actually gives you like the path coordinates, the base position. It's small because of high resolution screen, but it shows you the base position of the nodes happening. So it provides this kind of interactive visualization. And there's another one called Warograph. These are from Christian Fisher, who's at UTHSC. And so this is like a zoomable version of AlGViz, and it goes down the base level. And you can see the SNPs, in fact, up here at the top. It needs to go all the way where you can see the characters, but that's an open. Anyway, there's work going on it to make that possible. And so these are like these 2 visualization modalities that we're working on. And so then the other thing is, okay, so we said, why wasn't there an inversion in those genomes, right? And it was because it was the wrong generating sessions, because there are many assemblies of these strains. So we got the generated sessions from the same one that was in the paper that Ryan showed. And here you do see the inversion. It's right here. And you can see that a little more easily in, well, this is showing this loop. Yeah. And this is the inversion view in PGDB. It's showing the relative orientation of the paths to the graph. And so here, one is red, one is black, and something is there in different relative orientations. So, in fact, we were able to just detect it out of the box. Which it's under unit. And then I can show you some other quick things too. Just maybe the last example. So this isn't done by default, but this is just a tool for looking at the pairwise alignment between the 2. And So this is showing pairwise alignment that seems to overlap. So it matches. Yeah.
Why was the inversion not, why could it, what should it?
Different assemblies.
Yeah, I should mention. So I made a mistake, an embarrassing one yesterday by not having correct accession for a particular strain to correct, and it turns out that okay, well, some strain has inversion, some strains do not. And embarrassing for somebody who's working at the Microbiology Institute.
But also explain why the meaning graph one was completely linear.
However,
Yes. Yeah.
I run meaning graph again on the correct accession, and the insertion is still not found.
Yeah.
Competition.
It's found in
I'm surprised it doesn't find it though. But I guess when things diverge too far, right? Just
I think it's due to the size of the
size of it. Yeah. Okay, well that's all I have. I mean, we could do a longer practical, but it sounds like given the time, it's better to get a lunch and
Yes,
we have a discussion.
we agreed that tomorrow Erik is going to go on and continue the talk. I mean,
Look, we can have more of a discussion maybe.
yes,
tomorrow, yeah.
discussion maybe also you have something more.
Yeah, we can
Let's
do
like.
an
So,
interactive thing.
But
that would be good.
yes,
See more.
but given the time, I think it's better to have lunch and then if you have further questions.